Hello, everyone, and welcome to today's webinar titled KAPA HyperPETE, a novel primer extension target enrichment solution for rapid and reliable variant detection results. And this webinar is part of the 10th Annual Genetics Virtual Week. And I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Roche Sequencing. For more information about Roche Sequencing, go to sequencing.roche.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar, or go to the lobby. I now present today's speaker, Brian Godwin, the Director at the Reagent and Assay Development at Roche Sequencing Solutions. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Brian, welcome. You may now begin your presentation.
Thank you to everybody who's attending my talk today. So I'll just get right into it. So what is KAPA HyperPETE? It's a novel target enrichment capture method. The capture is based on primer extension target enrichment. This is where that PETE name comes from. It uses shotgun libraries prepared from DNA or RNA as input into capture. It combines hybrid capture performance with the ease of use and turnaround time of amplicon workflows. It provides confident answers for challenging and precious samples, and it is a single-day workflow from nucleic acid to pooled libraries ready for sequencing. It's capable of detecting all major somatic variant classes, including single nucleotide variants, short insertions and deletions, copy number variants, microsatellite instability or MSI, fusion transcripts, both known and novel partners. Panel sizes range from 5 kb to 250 kb for DNA panels, with a 300 kb capture panel size, and RNA panels from 5 kb to 50 kb. We offer both a custom and catalog content, and it's integrated with Navify Mutation Caller, which is our cloud-based secondary data analysis solution. We offer fast custom panel ordering turnaround time for rapid deployment and testing in your lab, and we offer FFPET DNA extraction through to sequencing-ready libraries with integrated data analysis. So here I'll show you the workflow overview. We start with either DNA or RNA into the workflow. This goes into any one of our library prep methods. KAPA HyperPrep kit is used for any pre-fragmented DNA, including cell-free DNA. The KAPA Hyper Plus kit is used for unfragmented DNA, and this is typically in what I'm going to show you in our somatic testing. And RNA goes into our KAPA RNA HyperPrep kit. Libraries then go into our HyperPETE kit and workflow. And this is what we're talking about when we say it's a single day from nucleic acid to enriched and pooled libraries. These pooled libraries go into sequencing, and the sequencing output goes into our Navify Mutation Caller, which generates VCF files as well as enrichment metric files. So how does KAPA HyperPETE work? So here I'm showing the start of from shotgun libraries. I won't go into how these libraries are made. My assumption is that everybody is familiar with this. These shotgun libraries have adapters with unique dual indices, and you have the option of working with adapters with or without UMIs or unique molecular indices. What I'm showing today, it's all with UMIs because this is what I'm showing is our somatic workflows. So we start with our shotgun libraries. In blue, I'm showing the target molecule, and this is just using a generic gene A exon 10. We denote that. And then all off-target molecules are the various other colors. The first step is to take a biotinylated primer and extend that with DNA polymerase. Following this extension, we bind to streptavidin beads and wash away any of our off-target molecules. We add specificity by using a release primer hybridization that is extended, pushing off in a target-specific method any of our on-target molecules, where any off-target molecule that doesn't get that secondary release primer will stay on the beads. We collect these released products and put them into a PCR reaction where we amplify. This is followed by QC, pooling, sequencing, and analysis. Here I'm showing turnaround time comparisons of KAPA HyperPETE. So this is shown here in blue, comparing to hybridization capture. This is completed in 1 day, whereas hybridization capture with an overnight hybridization takes 2 days. Anchored multiplex and KAPA HyperPETE have very similar turnaround times. And lastly, although KAPA HyperPETE takes a little longer than amplicon workflows, one of the advantages is that to cover long contiguous regions, you can do this in one tube, where amplicon methods typically need to be split into multiple tubes. Here I'm showing that the KAPA HyperPETE application turnaround time is dependent on the different library prep methods. For somatic tissue DNA, where we use KAPA Hyper Plus, the total turnaround time is approximately 9 hours. For somatic cell-free DNA using KAPA Hyper Prep, it's about 8.5 hours. And then for RNA, where we use KAPA RNA Hyper Prep, it's about 9.5 hours. A key here is that the enrichment workflow is the same across all of these applications, with the only caveat that PCR cycling conditions after enrichment may be different. Now I'm going to show you some development data. This is actually coming from our verification studies, and I'm going to show you results from a few of our catalog somatic oncology panels. I'll start with somatic tissue DNA. Here I'm showing you our KAPA HyperPETE pan-cancer panel and gene list. This is all alphabetical order. Feel free to look through this list while this screen is up. The panel content includes 1,132 CDS regions across 86 oncogenes, covering single nucleotide variants, indels, and copy number variants. There are 190 MSI loci for characterizing MSI status of samples. This is denoted in purple. The capture panel size is approximately 300 kb. Samples used in this study include 16 adjacent normal FFPET samples of varying quality to assess enrichment performance metrics. There are also 4 xenograph FFPET samples run in duplicate to assess enrichment performance metrics and MSI calling performance. And then there are 4 FFPE format reference samples and 12 cell line reference samples run in duplicate from various sources to assess enrichment performance metrics and variant detection performance. So here I'm showing the experiment setup. FFPET DNA was extracted using the KAPA NGS DNA extraction kit, and quality was measured using the KAPA NGS FFPE DNA QC kit. Libraries were generated using the KAPA Hyper Plus kit in combination with a KAPA NGS FFPE DNA polishing kit and KAPA Universal UMI adapters with 10 nanograms, 50 nanograms, and input based on quality assessment. When we use that quality assessment, for low quality samples in this study, we use 60 nanograms to 248 nanograms of input. For mid-quality samples, it was 40 to 54 nanograms, and for high quality, it was 20 to 26 nanograms. Libraries were captured using the KAPA HyperPETE reagent kit and sequenced on an Illumina NextSeq 550 instrument. Total read pairs per sample range from 19 million to 55 million and were analyzed using the Navify mutation caller to assess enrichment and variant detection performance. Here I'm showing you the enrichment performance results, and we'll start with on-target rate up at the top here. So a few things to look at. On the far left is 10 nanograms of input, followed by 50 nanograms, and then quality adjusted input. The various qualities, low quality is in blue, mid quality is in orange, and high quality is in green. So on target range from 68 to 77% with a median of 73% across all sample qualities and input amounts. And this is all on the top here. Quality adjusted input over here on the right, percent dedupe panel exon bases greater than 300x here. All of the samples were greater than 98%. And for 500x, all samples were over 93% and perform well across all sample qualities. The reason why this is important is I want to show that using the quality adjusted input will give you the most reliable result in your enrichment. In the middle, I'm showing 50 nanograms of input. Here, the percent dedupe panel exon bases greater than 300x had a minimum of 92% in the low quality samples, and at 500x, the minimum was 75%. So the performance is still good here, but there is slightly more variability when using 50 nanograms in comparison to the quality adjusted input. At 10 nanograms on the far left, you can see that there's more variability, and this is dependent on the sample quality the sample quality input. So you can see low quality ranges much lower than mid quality and high quality samples for 300x, and this is even more pronounced at the 500x level. One thing that I'm not showing here, but I'd like to make a mention of, results were comparable when downsampling to 27.5 million read pairs. And the reason why I want to point this out is this is the guidance that we have in our instructions for use to target about that many read pairs. So I just want to make sure to point that out, that the results are comparable when using what we're giving as our guidance. Here I'm showing variant detection performance. So this top table is including SNVs and indels, or all of the short variants. On the left shows each of the samples that were used. Here I'm showing the input amount, and the take home is that there's a true positive rate of 100% across all of these reference samples and different input amounts. For copy number alterations or copy number variants, we see a true positive rate for all of the samples and input amounts in this table. And lastly, MSI status or MSI high status for these samples, we detected 100% of all of the MSI status in these samples. Moving on to somatic cell-free DNA. Here I'm showing the KAPA HyperPETE hotspot panel, which is a much smaller panel than our pan-cancer panel. This includes 166 CDS regions across 37 oncogenes covering SNVs and indels in the cell-free DNA configuration. The capture panel size is approximately 37 kb. I'm also going to show you results using the pan-cancer panel contents, which I showed you earlier. One of the keys here is we're only detecting single nucleotide variants and indels in this particular configuration. The CNVs and the MSI status are not detected in this particular application type. Capture panel size, again, is approximately 300 kb. Samples used in this study include 4 Sera-seq ctDNA mutation mix samples run in duplicate to assess enrichment performance metrics and variant detection performance. There are also 16 healthy donor plasma DNA samples used to assess enrichment performance metrics. The experiment setup, libraries were generated using the KAPA HyperPrep kit in combination with the KAPA Universal UMI adapters with either 10 nanograms or 50 nanograms of plasma cell-free DNA or those fragmented reference cell line samples as input. Libraries were captured using the KAPA HyperPETE reagent kit and sequenced on an Illumina NextSeq 550 instrument. Total read pairs per sample range from 6 million to 14 million for the hotspot panel and 55 to 81 million for the pan-cancer panel, and these were analyzed using the Navify mutation caller to assess enrichment and variant detection performance. Here I'm showing that enrichment performance. So for the hotspot panel, which is on the left, and the pan-cancer on the right, and 10 nanograms in blue, 50 nanograms in orange. On-target rate for the hotspot panel over here ranged from 65 to 73%, and it was 70 to 73% for the pan-cancer panel. We have good performance across all sample input amounts and types in this case. At 50 nanograms of input, or in the orange, the percent dedupe panel exon base is greater than 1000x. Here, for the hotspot panel, was greater than 99%. And for the pan-cancer panel, it ranged from 97 to 98%. At 3000x, these numbers are a little bit lower, but that's because we're looking at 3000x. The hotspot panel ranged from 80 to 94%, and the pan-cancer panel ranged between 67 and 85%. At 10 nanograms of input in blue, the percent dedupe panel exon base is greater than 1000x for the hotspot panel ranged from 83 to 99%. And for the pan-cancer panel, it was 80 to 94%. So this is not unexpected to see that the 10 nanogram input amount is going to perform a little worse than the 50 nanogram input amount. Just to note, at 3000x, the performance using 10 nanograms input is actually lower, but this is something that you should expect because there are only approximately 3000 genome equivalents in 10 nanograms of input. So this is as expected. For variant detection performance, 100% of short variants, including SNVs and indels, at the 1% allele frequency were detected across all reference cell line DNA samples at both input amounts in these panels. So this is down in this table. This is the hotspot panel at 1% allele frequency, 100% true positive rate. In pan-cancer, the same thing, 100% true positive rate. At 50 nanograms of input for 0.5 allele frequency, down here, there's a 100% true positive rate for the hotspot panel and 98.4% true positive rate for the pan-cancer panel. For 10 nanograms of input at the 0.5% allele frequency, the hotspot panel has a true positive rate of 94%, and the pan-cancer panel has a true positive rate of 96.9%. Something to note is that all the variants that were missed in the caller were present, but read support was lower than the cutoff used in the analysis pipeline. It's important to know that the analysis pipeline is customizable, so you can change this default parameter to something that is you can be satisfied with in your lab. For somatic tissue RNA, here I'm showing the KAPA HyperPETE lung cancer fusion panel. This is the gene and exon list. The header of this includes each of the genes that are in this panel, and below that are all of the exons that are covered in this panel. The panel content includes 16 oncogenes covering 137 exons, designed to detect both known and novel fusion partner transcripts at both the 5' and 3' junctions of the exons. There are 4 housekeeping genes which serve as positive enrichment controls in the workflow, and I'm just showing you what these genes are here in this line. Samples used in this study include 2 cell line samples run in duplicate and 14 normal adjacent FFPET samples to assess enrichment performance metrics. And there are 2 Sera-seq RNA fusion FFPE and 1 Horizon Diagnostics RNA fusion FFPE sample. Each of these are run in duplicate to assess enrichment performance metrics and variant detection performance. The experiment setup includes RNA quality was determined with the DV200 score using the Agilent RNA 6000 Pico assay on the bioanalyzer. Libraries were generated using the KAPA RNA HyperPrep kit in combination with the KAPA universal UMI adapters with either 10 nanograms or 50 nanograms of RNA while adjusting PCR cycles based on the input amount and the DV200 score. Libraries were captured using the KAPA HyperPETE reagent kit and sequenced on an Illumina NextSeq 550 instrument. Total read pairs per sample range from 3.6 million to 17 million and analyzed using the Navify mutation caller to assess enrichment and variant detection performance. Enrichment performance results are on the left. Following rRNA read removal, 0.8% to 11%, which made up 0.8% to 11% of all reads, On-target rate was 92% to 97%, with good performance across all sample input amounts, qualities, and types. So similar to the FFPET DNA, blue are low-quality samples, orange are mid-quality samples, and green are high-quality samples. And then both these input amounts are shown here. Housekeeping targets, which are used as the enrichment controls, as I talked about earlier, are considered as on-target reads and make up between 26% and 70% of reads, depending on the sample type. So this data is not shown. This is something that we expect, and we think that it's very important to use these housekeeping targets as a positive enrichment control. Here I'm showing variant detection performance. So this is very easy to present. So 100% of fusions in the FFPE reference RNA samples were detected at both the 10 nanogram and 50 nanogram input amounts. Comparable variant detection results were achieved when downsampling to 1 million read pairs. So I don't show that data here, but the data looks basically the same as it does in this particular table. This is important because this is the actual sequencing depth guidance that we give in our instructions for use, or our IFUs. Something to note is that the EGFR Sept14 variant in the Cereseq Fusion RNA Mix V4 was manually curated as the Navify mutation caller identified a different EGFR partner that has an homologous sequence to Sept14. So this is something that we've seen in our pipeline. We know it's just a matter of the caller selecting a particular target, when in fact it's just the same sequence across multiple targets. With that, the KAPA HyperPETE workflow and reagent kit provides both a fixed and customizable content target enrichment solution for reliable detection of all major somatic variant types, including single nucleotide variants, insertions and deletions, copy number variants, novel fusion transcript partners, and MSI status. It's a fast and easy workflow that generates sequencer-ready libraries in a single day when starting from purified nucleic acid. It's a novel primer extension target enrichment solution that provides performance equivalent to overnight hybridization capture technologies. And it shows flexibility in nucleic acid input amounts from 10 nanograms to 50 nanograms, or input based on quality adjusted input, or on quality adjusted QCs. Sample types include high quality DNA, plasma DNA, FFPET DNA of varying qualities, and FFPET RNA of varying qualities. And with that, I'd like to thank you for your time, and I'm happy to answer questions.
Thank you so much, Brian, for your time today and for your important research. And before we go, I want to thank our audience for joining us today. I'd also like to thank our sponsor, Roche Sequencing, for sponsoring today's webinar. Questions submitted today and on the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed up to 2 years until April 21st of 2024. Labroots will alert you via email when it's available to replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care. Bye-bye.